Hello students, today we are going to continue with this chapter, that is sexual reproduction in flowering plants. Okay, so in the last session we started this chapter and I taught you about LS of a bisexual flower. We said about different parts of a bisexual flower, and then I told you we'll be starting to study about the pre-fertilization events. So we started to study about androecium first. So I taught you about the male reproductive structure, that is stamen. Okay, so we studied about TS of young anther in the last class. Okay, so when you just recall that, okay, so just think about the structure. We are moving on with the next part, that is TS of mature anther. Okay, so in TS of young anther, just recall, I told you there are 4 compartments. Okay, that is called as microsporangia, and it is covered by 4 layers of cells. So here today what we are going to study is how the anther looks, that is TS of mature anther. So there is a lot of difference between young anther and mature anther. There are so many changes which has happened. Okay, so today we are going to study about TS of mature anther. So this also is a probable 5 marks question. Okay, the diagram of TS of mature anther. So the layers and all the names, I hope you can remember, it's the same. So just have a look at the diagram here. This is the structure of TS of mature anther. Okay, so here also you see the outermost layer, which is called epidermis. Next to that layer, endothecium. And then inner to that is few layered, it may be 2 or 3 a few layered cells, which we call as the layer is called as middle layer, and next to that is a single layered structure called as tapetum. And I told you tapetum is a tissue which has a lot of reserved food materials. Okay, it's a nourishing tissue. So this 3, epidermis, endothecium and the middle layer, it is just giving the protection. Okay, so one more main difference what you can find from this one, that is TS of mature anther and young anther, you just compare it, you see that here it is open. Okay, so we call it as dehisced anther. So when anther matures, you will see that it has to break open because it has to release the pollen grains, right? So here you will see that it is open. Okay, so this is called as dehisced anther. And here if you see, there is a single compartment in each lobe, and this we call it as pollen sac. Now we don't call it as microsporangia, we call it as pollen sac. So pollen sac contains pollen grains. So in the microsporangia, if you recall, I told you there is a mass of tissue called as sporogenous tissue, okay, which has developed into pollen grains. So you will see pollen grains in these pollen sac. Okay, so this will be the structure of matured anther, matured dehisced anther. So you can see the pollen grains are coming out. Okay, so this is a diagram which you need to draw for a 5 marks question. Okay, so the changes you just need to compare with the young anther and compare it to this, you will see the difference. Okay, so no microsporangia here, it's a pollen sac which is filled with pollen grains, and it is open, that is it is dehisced anther, that is to release the pollen grains. Okay, so this is the structure of TS of mature dehisced anther. Okay, we studied about the structure, fine. Now what we have to know is, now I told you sporogenous tissue has been converted into pollen grains. So how did that transformation happen? How did those changes happen? We will have to study now. Okay, so what we have to study now is microsporogenesis. Okay, so what is microsporogenesis? Now, microspore mother cell. Now, what is this microspore mother cell? So, I told you within the tissue that is within the microsporangium, there is a tissue called as sporogenous tissue, right? So, this sporogenous tissue, the cells we call it as microspore mother cell. Now, this microspore mother cell will be undergoing cell division to form microspores. Okay, so what type of cell division happens there? It is meiosis. So, microspore mother cell undergo meiosis to produce microspores, which is called as microsporogenesis. Okay, so this microsporogenesis is the production of microspore from microspore mother cell through meiosis. Now, this microspore mother cell can be also called as pollen mother cell. Okay, we can also call it as pollen mother cell. So, either of these two terms you can use. So, pollen mother cell or microspore mother cell. So, what do you think? Why does meiosis happen? What is the reason? So, I just told about this in the previous session also. Pollen grains, that is, now it is meiosis is happening and formation of microspore happens. Now, these microspores are developing into pollen grains. Now, you know, pollen grains are carrying male gametes. Okay, so you know gametes are always haploid. Okay, so now microspore mother cell is diploid. Okay, if this has to produce a haploid cell, it has to undergo meiosis. Therefore, meiosis happens and microspores are formed. Now, this microspore is developing into pollen grains. Okay, but only this part, that is microspore mother cell undergoing meiosis and it producing microspores. This much of development we call it as microsporogenesis. So now I told you about now what is microsporogenesis. Only one part of the development we call it as microsporogenesis. That is only microspore mother cell undergoing meiosis and producing the what is microsporogenesis. Okay, so now the whole process we are going to study. So microsporogenesis is only a part of the development, that is only microspore mother cell undergoing meiosis and it produces microspores. So only that part of development we call it as microsporogenesis. So now we will see in detail. Now microspore mother cell, okay, I told you it can be also called as pollen mother cell. That is nothing but the tissue what we find inside the microsporangium, what we call as sporogenous tissue. That's those cells. Every cell is capable of dividing and producing the pollen grains. So this microspore mother cell is undergoing meiosis. And you know that the product of meiosis is 4 cells. So you know one parent cell undergoes meiosis, okay, and it will produce 4 daughter cells. So I hope you can just recall that we had studied that in the previous year's chapter about mitosis and meiosis. So meiosis is one diploid cell undergoing cell division and it produces 4 daughter cells. I hope you can just recall that. So here also, since this cell is undergoing meiosis, microspores which are formed, you know that it is actually 4 cells together. Okay, so it's a bunch of like 4 cells which we call as microspore tetrad. So 4 microspores are produced from one microspore mother cell. Now this microspore tetrad, okay, so it is like 4 cells together. So immediately they don't separate from each other, they are together, which we call as microspore tetrad. As the anther matures, okay, so as it dehydrates, so you know that the anther wall has to break open. So dehydration takes place there, that is it dries up. So along with that you will see that microspore tetrad, that is 4 microspores together, they will separate from each other and form the microspores, and this microspore will be developing into pollen grains. So I hope you're clear about that one. So only development from here to here we call it as microsporogenesis. So this is in detail what I have told you, what exactly happens during microsporogenesis. So microspore mother cell undergoing meiosis, it forms a cluster of cell, that is cluster of 4 cell, which we call as microspore tetrad. For all the 4 cells will dissociate from each other, separate from each other as the anther matures, and each microspore is developing into pollen grains. This is how the whole development goes about. So that we are trying to explain with the diagram here. Okay, so you can see microspore tetrad. Okay, so I told you this is a structure of microspore tetrad. So you can see 4 cells here, that is nothing but 4 microspores which are formed initially, all 4 are together, and that is called microspore tetrad. Now after there is these 4 cells, as I told, as the anther matures it will separate. So out of these 4 cells, 1 cell have been shown here. This is a single microspore. And I told you microspore will be developing into pollen grain. So that, how does that development take place, we will see with these diagrams. Okay, so this is microspore tetrad, 1 cell from this, that is microspore, which is developing into pollen grain. So you will see that initially, okay, the nucleus enlarges, lot of reserved food materials, you can see the production of vacuoles also. Okay, so it prepares itself for cell division. Okay, that is microspore is preparing for cell division. Now here you will see that asymmetric spindles are formed. Okay, so cell division happens, it's an unequal division what happens, and you should know that this microspore is a haploid cell now. Because it's a product of meiosis, microspore is a haploid cell. Now this cell is undergoing cell division, means it is undergoing mitotic division. So mitosis happens, that is unequal division happens. So you can see here asymmetric spindles. So unequal division happens, and you can see that 2 cells are formed which are of 2 different size. One is a large cell, another is a smaller cell. Okay, so this is a pollen grain. So microspore has developed into pollen grain. So if you see the pollen grain structure here, it has 2 cells. So 2 cells are of 2 different size, that is a result of unequal division. So you should know that the larger cell, which is called as vegetative cell, and the smaller cell is called as generative cell. Okay, so this is how a microspore mother cell undergoes meiosis. It produces microspore tetrad, and each microspore in this way develops into pollen grain. Okay, so this developmental stages, see stages of a microspore maturing into a pollen grain. So this can also be asked as a 3-mark question. Okay, so then you need to draw this diagram, and you just need to explain how a microspore develops into pollen grain. I have explained about the whole process of development of pollen grain, that is microsporogenesis, and how the pollen grain develops. Okay, so the whole process we have studied. Now we will concentrate on the structure of pollen grain. Okay, so pollen grain, how this develops, I've explained now. Okay, so we'll concentrate on the structure. Now when we say pollen grain, see, you take out any flower. Okay, so hibiscus flower is the best one for this. You just open out the flower and you just touch the stamen part, you get some powdery structures over to your hand, yellowish powdery structure. So that is nothing but the pollen grains. Okay, and there are like varieties, each species of plant produces different types of pollen grains, that is the structure, the external morphology, that is the structure, it varies. And if you see, like, we can collect the pollen grains over the slide, okay, putting a drop of water, we can collect it and we can observe it under the microscope. You will see that there are so many varieties, the color of the pollen grains, the shape, the architecture, everything is so different from species to species. Okay, but overall we'll just concentrate on the structure of each pollen grain now. So this will be the structure of a matured pollen grain. So I told you that pollen grain contains two cells, one larger cell and one smaller cell. Okay, so now each part we will study. So pollen grain is covered by two layers. Okay, it has an outer layer which is called exine. Okay, so exine is the outermost layer of pollen grain. Now this exine is made up of a material which is called sporopollenin. Okay, so it is made up of a material called as sporopollenin. Okay, and this is one of the hardest material. Okay, so the hardest organic material which we find on the earth, and there is we say that the sporopollenin is very resistant to high temperature, strong acids or alkali. Okay, so till now we say that there is no enzyme, okay, which can degrade this sporopollenin. So it is a very hard material. So this hard material is the outermost layer of the pollen grain. Okay, so exine is made up of sporopollenin. Okay, so sporopollenin is a material, okay, where the exine is made up of. So this exine is a very hard layer, and I told you sporopollenin, the characteristic feature of that also I told that it's very hard, where no enzymes can degrade it. Okay, therefore pollen grains, we find them as fossils. Okay, so in the earth's crust, the plants which we were living almost so many thousands of years back, okay, the pollen grains have been excavated. So in the soil we do find the pollen grains, we say fossils. Okay, so within the earth's crust, different layers of earth's crust, we see that the sporopollen material is still without decomposition. We find it like that. So pollen grains can be fossilized only because the exine is made up of this sporopollenin. Okay, so that is about the exine. And you should know one more thing, it is not a continuous layer. Okay, so exine is not a continuous layer. In between you will see some pores. Okay, so that pores we call it as germ pores. Okay, so sporopollenin is a discontinuous layer where here and there you will find some apertures or the openings which we call as germ pores. Okay, so presence of germ pores in the exine layer. Then moving on to the next layer, next to this, the inner layer, which we call as intine. Okay, so intine is the second layer, and intine is a soft layer which is made up of cellulose and pectin. Okay, so that is intine. So as I told you, we see two cells, vegetative cell and generative cell. So the cytoplasm of the pollen grain, it is covered by plasma membrane as usual. Okay, and we find two cells. Larger cell is called as vegetative cell and smaller cell is called as generative cell. Okay, now this vegetative cell, it's a large cell with irregular shaped nucleus with lot of reserved food materials and all. This generative cell is a small cell, spindle shaped cell, which actually floats in the cytoplasm of the vegetative cell. Okay, and these are the two cells, vegetative cell and generative cell. So this one, that is the structure of a matured pollen grain, can be asked as a 3 marks question. Okay, then you need to draw this diagram, okay, with all the labelings. So exine made up of sporopollenin, intine soft layer made up of cellulose and pectin, and it has two cells, one large cell, vegetative cell, and one smaller cell called as generative cell. Okay, so that is about the structure of the pollen grain. Now you should know that I told you pollen grains are released from anther. So we studied about that just now, that anthers will dehisce, it will break open and it will release the pollen grains. Now almost 60% of angiosperms are releasing the pollen grains in this stage only. That is, we call this stage as two-celled stage. Okay, and we say 60% of the angiosperms, okay, 60% of the angiosperms are shedding the pollen grains or releasing the pollen grains at this stage only, which we call as two-celled stage. What about the remaining species of angiosperms? So we say remaining 40% of the angiosperms are releasing the pollen grains in three-celled stage. Okay, so they are releasing in three cells. Now what is this three-cell stage? Because I told you pollen grains having two cells, vegetative one cell, generative the other cell. Okay, but you should also know that this three-celled stage, okay, that is nothing but generative cell, okay, will again divide and form two cells. So two generative cells will be formed before the pollen grains are released. Okay, so the remaining 40% of the angiosperms are releasing pollen grains in three-cell stage. So that is, it will have one vegetative cell and two generative cell. So one mitotic division happens here, and one generative cell is dividing into two generative cell. And you should also know that the nucleus of generative cell is nothing but they are the male gametes. Okay, so then what doubt arises is, how come some species are having two nucleus in the generative cell, some species are having only one? So how is that? So you should know that even in a two-cell stage, what we see, this division do happen, but it happens later, not during the shedding of pollen. Okay, so pollens are shed at two-cell stage, but this division will happen but in the later stage. In some, as I told, in the remaining 40%, okay, so remaining 40% of the angiosperms are shedding the pollen grains at three-cell stage. Here the division has happened early, that's it. Okay, so that's the only difference. So that is about the structure of pollen grains and also about the different stages of like how they are released, either in three-cell stage or two-cell stage. So that is about the structure of pollen grain. Now we should also know like how are these pollen grains influencing human beings. Okay, so some pollen grains, that is pollen grains of some species of plants, you will see that it is causing allergy to human beings. Okay, so you might be aware of some species will cause allergy, and one main plant what comes to our mind is Parthenium. Okay, so Parthenium plant produces, that is, it releases the pollen grains, and those pollen grains are very allergic. So we just inhale that and we get all different types of allergic symptoms. Okay, so allergy, it depends on like for how long you are exposed. Now for example, we just exposed for a little bit of time, maybe you get temporary symptoms like maybe you start sneezing or you get headache or something like that. But for some people who are like very sensitive to these pollen grains and all, what happens is if they are exposed for a longer time, so for example maybe there are some plants around your houses which are releasing these pollen grains and you are continuously inhaling that and all, then it may cause some breathing disorders, respiratory tract disorders like asthma, bronchitis. Okay, so all these disorders can be caused because of the pollen grains of certain species of plants, not all the species, certain species. Now we just spoke about the negative part. We also should know about the positive things about the pollen grains. Okay, so pollen grains of many species of plants, they are very nutritious. Okay, so it has become like a fashion of taking this a supplement, pollen supplement, especially in the western countries. We find that they get different products of pollen grains like pollen tablets, pollen syrups and all they get, and they take this supplement, especially athletes. Okay, and race horses also are given these supplements, and it is seen that when they consume that, their performance increases or performance becomes good. Okay, so because it's affecting their performance in a positive way, they have the habit, it is mostly in the western countries, it is they take the supplement of pollen grains. As I told, different products are available. Okay, so that was about the positive part, like how we human beings are influenced, negative and positive, both the things I told you now. Okay, so that is about the effect of these pollen grains to human beings. Now we'll come back to the plants now. So in plants, I told you now pollen grains at any stage, 2-cell stage or 3-cell stage, it is released from the anther. Now once it is released, what next? It has to go and settle on stigma, because you know this is carrying male gametes. Pollen grains are carrying male gametes, and you know that it has to land on stigma, that is fertilization should happen, right? So from the time it is released from the anther, you will see that we say or in a simple words we can say the lifespan of pollen grains. So pollen grains, from the time it is released, okay, we say viability of pollen grains, that is the lifespan of pollen grains. It has to land on stigma before it loses the viability. Okay, so what is this viability? So I told you it's nothing but, in simple words we can say like a lifespan of this pollen grain. So for how long they can be active, for how long they can bring about fertilization, we call it as viability. So before a pollen grain loses its viability, it has to go land on stigma and it has to bring about fertilization. So how long is the viability of the pollen grains? So viability we cannot say precisely, because it depends on from species to species. Now for example, some cereals like rice and all, the pollen grains once they are released, their viability is only 30 minutes. So within this 30 minutes, the pollen grain has to reach the stigma and it has to bring about fertilization. Okay, so after that it will lose its viability, means it will not be able to bring about fertilization. Okay, and some examples like leguminosae, solanaceae. Okay, there are some members where the viability of pollen grains is it's like many months. Okay, so when we say many months, it is like pollen grains released today, it remains viable till 1 month, 2 months or even 3 months. Okay, so now see, for some it is only minutes, 30 minutes, in some species it is months. So viability of pollen grains, you will see that it is not, it depends from species to species. So before it loses its viability, it has to land on the stigma and bring about fertilization. Okay, now we also have heard about sperm bank, okay, in animals and even in human beings also, for bringing about artificial insemination. Okay, so where the sperms or the semen is collected and it is stored. In the same way, these pollen grains can also be stored. Okay, so it can be stored in liquid nitrogen. Okay, so storage of the pollen grain is we can do it in liquid nitrogen at -196 degrees Celsius. So this is a condition where the pollen grains are stored. So pollen grains can be stored in liquid nitrogen under -196 degrees Celsius. So same like the sperm bank, okay, we also find pollen bank where pollen grains can be stored. Okay, so this is the end of androecium. So we started to study about the structure of the male reproductive structure, that is anther. We studied the internal structure, how young anther looks like, how the matured anther, what are the transformation happening there, how the pollen grains are produced, and everything we studied. Okay, so with that we finished the androecium part. Okay, so after this we have to move on with gynoecium, that is the female reproductive structure. Okay, so with that, after completing that, we'll be moving on with the whole process of sexual reproduction. Okay, so we'll stop at this point for this session. So gynoecium, I'll be starting in the next session. So today, what I have explained, I want you all to draw two diagrams. One is TS of mature anther, I told you, which is a 5 marks question. Okay, and pollen grains. Okay, the whole series of diagrams showing the development of pollen grains. I want you people to draw these two diagrams, practice it. Okay, so I told you the last session also that below the video there is a link which is given. You can open that link, take a pic of the diagram which you have practiced in your book, and you can post it. Okay, so we'll meet in the next session. Thank you. Have a good day.